Let's begin with something deceptively simple. Every species of living organism, whether it's a bacterium, a baboon, or even your backyard blueberry bush, has evolved from a different ancestral species. It might seem like life today is frozen in time, going about its usual business, unchanged. But in reality, evolution hasn't hit the pause button. It's still humming along quietly in the background. Evolution, as a concept, isn't just a chapter in a biology textbook. It's the spine of the whole subject. It gives biologists a way to frame their questions, a lens through which the tangled web of life starts to make some sense. What makes it so compelling is that it works. Time after time, it gives us predictions about how life behaves, changes, and adapts. And those predictions hold up under scrutiny. There's a quote that tends to come up in every biology classroom at some point. Nothing in biology makes sense except in the light of evolution. That was said by a geneticist with a flair for cutting to the point. And he was right. Evolution isn't just a side note, it's the unifying thread. The idea that all life shares a common ancestry and is gradually diversified over time. Without it, biology is just a big, fascinating pile of facts. With it, the pile becomes a story. Charles Darwin is most famously associated with the idea of natural selection. But he wasn't entirely alone in coming up with it. In the mid-1800s, both Darwin and another naturalist, Alfred Russel Wallace, independently landed on the same fundamental insight about how evolution works. Interestingly, they didn't do it in armchairs or laboratories. They did it in the field, knee-deep in the wild. Darwin spent 5 years circumnavigating the globe aboard a ship called the HMS Beagle, making stops in places like South America, Australia, and Southern Africa. Wallace, not one to be outdone, spent years exploring the Amazon and later the sprawling islands of the Malay archipelago. Both were collecting specimens, making notes, and slowly piecing together a picture of how species might change over time. Darwin's visit to the Galapagos Islands turned out to be especially pivotal. There he noticed that certain animals, like finches, looked broadly similar but had slight variations depending on which island they called home. The differences weren't dramatic, but they were purposeful. One finch might have a thick, heavy beak made for cracking seeds, while another just a few miles away had a slender beak better suited to plucking insects from tree bark. These weren't just quirks of nature. They were clues. Wallace made similar observations during his travels. Across different regions, species seemed finely tuned to their environments. Eventually, both he and Darwin reached the same conclusion. Nature was selecting for traits that helped individuals survive and reproduce. Over time, these traits became more common. This is what we now call natural selection, though at the time, the phrase survival of the fittest also started making the rounds. A bit dramatic, but it stuck. Darwin illustrated this idea with an example from those same Galapagos Islands. This time, the tortoises. On some islands, food was harder to reach, especially during dry spells. Tortoises with slightly longer necks could stretch just a bit farther to nibble on higher vegetation. Those with shorter necks, unfortunately, didn't fare as well. Over generations, the population shifted. Long necks became the norm, not because the tortoises were trying to evolve, but simply because the ones with the useful trait left more descendants behind. Darwin's idea of natural selection, while revolutionary, was built on 3 principles that are remarkably simple. First, most traits are inherited. Offspring tend to resemble their parents, not exactly, but closely enough. Now, back in Darwin's day, no one really knew how inheritance worked. Genes hadn't been discovered, and DNA wasn't even a concept. Still, people had noticed that traits seemed to run in families, whether you were a pea plant or a pigeon. Second, more offspring are produced than the environment can possibly support. This means there's competition for food, for space, for sunlight, for every little advantage. Darwin didn't invent this idea himself. He borrowed it, quite sensibly, from the economist Thomas Malthus, who had warned that human populations grow faster than the food supply. Nature, it turns out, has the same problem. Third, there's variation. Not every individual in a species is a carbon copy of the next. Some are a bit taller, a bit faster, a bit tougher, or just slightly luckier when it comes to timing. Many of these differences are inherited, which brings us back to Principle 1. If a certain variation gives an individual an edge in survival or reproduction, that trait is more likely to be passed on. Over time, those traits build up in the population. That's evolution by natural selection, or as Darwin put it, descent with modification. It's a slow process most of the time. So slow, in fact, that when Darwin and Wallace presented their ideas in 1858 to the Linnean Society in London, the audience found them underwhelming. No fireworks, no scandal. Just two papers quietly read out loud, laying the groundwork for a seismic shift in science. A year later, Darwin published On the Origin of Species, and the world started paying attention. Now actually, watching natural selection happen is tricky. It's not the kind of thing that puts on a show, but sometimes nature cooperates. In the 1970s, two researchers, Peter and Rosemary Grant, began studying finches on a little island in the Galapagos called Daphne Major. These were the same finches that had caught Darwin's eye more than a century earlier. The Grants noticed that beak size varied among the birds, and these differences were inherited. When an El Niño year brought heavy rains, the kinds of seeds available shifted. Big, tough seeds became scarce. Softer seeds became more common. Suddenly, birds with smaller, nimbler beaks had the upper hand. They ate better, survived better, and passed on those smaller beaks. Measurements over time showed the whole population's average beak size shrinking. Not by much, but enough to notice. Then, when the environment shifted back, so did the trend. Natural selection, as elegant as it is, can't work without something to work on. It depends entirely on variation, differences between individuals in a population, but not just any differences. For natural selection to do its quiet sculpting over generations, those differences must have a genetic basis. If a trait isn't inherited, it won't be passed on, no matter how useful it might be. Take height, for example. One person might be taller than another because of their genes, or simply because they ate more vegetables growing up. Natural selection only concerns itself with the former, the heritable part. So where does this genetic variation come from? Two main sources: mutation and sexual reproduction. A mutation is just a change in DNA, a tweak, an error, a happy accident, or sometimes an unfortunate one. Mutations are the raw material of evolution. They create new alleles, different versions of genes, and with them new traits. Some mutations are helpful, some are harmful, and many do absolutely nothing. They sit quietly in the genome, neither boosting survival nor dragging it down. But every so often, a mutation provides an advantage, and that's where natural selection perks up. Then there's sexual reproduction. Every time two parents contribute their genetic material to create offspring, it results in a genetic reshuffling. Like shuffling a deck of cards, it produces combinations that have never existed before, new arrangements of old traits. This is why no two siblings, except identical twins, are exactly alike, and why evolution has so much raw material to work with. When a heritable trait gives an organism a better shot at survival or reproduction in its environment, we call that an adaptation. And over time, if enough individuals in a population share that trait, we say the group has become adapted to its environment. Think of the webbed feet of platypuses, ideal for paddling through water, the thick fur of snow leopards, perfect for mountain chill, or the cheetah's sheer speed, which really puts the pressure on prey animals to keep up, or more realistically to hide well. But remember, whether a trait is good depends entirely on the environment. What works well in one setting might be a liability in another. Imagine a plant that thrives in a rainy tropical forest with large sun-catching leaves. If the climate dries out, those big leaves suddenly become a burden. They lose too much water. In this new environment, plants with smaller leaves fare better, and natural selection takes a new direction. This is how species evolve, not in leaps and bounds, but in subtle, gradual shifts. Over time, these shifts can produce striking changes in form and function. Sometimes species that share a common ancestor evolve in very different directions, adapting to very different lifestyles. That's known as divergent evolution. One group of flowering plants might develop tall stalks and wind-dispersed pollen, while another ends up with bright petals and nectar to attract insects, all from the same starting point. In contrast, convergent evolution describes a different pattern. Here, unrelated species evolve similar traits, not because of shared ancestry, but because they face similar challenges. Bats and insects both fly, both have wings, but they arrived at flight via very different evolutionary routes. Their wings may look alike, but under the surface, the blueprints are completely different. These changes, big or small, happen over deep time. Natural selection acts on individuals in the present, but its full effects take generations to unfold, sometimes thousands, sometimes millions of years. Yet it's this slow process that explains the vast variety of life we see around us. Now, when Darwin and Wallace proposed their theory of natural selection, one piece of the puzzle was still missing: how traits were actually passed down. At the time, many believed in blending inheritance, the idea that offspring were simply an average of their parents, like mixing paint. The trouble is, blending would dilute advantageous traits, not preserve them. It made evolution look impossible. Unbeknownst to Darwin, a monk named Gregor Mendel had already conducted experiments that showed inheritance didn't work that way. Through careful studies of pea plants, Mendel discovered that traits are passed down in discrete units, what we now call genes. His work was published in the 1860s, shortly after On the Origin of Species. Unfortunately, it gathered dust for decades. It wasn't until the early 20th century that scientists revisited Mendel's findings and realized that genetics and natural selection weren't at odds. They were two halves of the same explanation. This merging of Darwin's theory with Mendel's principles gave rise to what we now call the modern synthesis. The modern synthesis united evolutionary biology with genetics. It showed how natural selection could act on genetic variation to shape populations over time, connecting the small-scale changes we see within populations, or microevolution, with the large-scale changes that lead to new species and even entirely new groups. Macroevolution, at every level of life, from tiny molecules to vast ecosystems, we can trace the quiet fingerprints of evolution. Even in the earliest days of the theory, Charles Darwin spent countless hours peering into nature's patterns, seeing in them not chaos, but a deep, underlying logic. He saw hints of a story that stretched far back into time, a story of change, adaptation, and shared ancestry. One of the clearest windows into that story is the fossil record. Fossils, those stone-cast remains of once living things, tell us that the organisms of the past were often quite different from the ones we see today. By dating these fossils and organizing them in layers, deeper fossils being older, shallower ones more recent, scientists have built a rough timeline of life on Earth. And across that timeline, the story becomes visible. Horses, once the size of house cats, gradually evolving into the tall grazers we know now. Early human ancestors walking upright, their tools growing more refined over time. Another line of evidence comes from what we call homologous structures. These are physical features shared across different species, not because they serve the same purpose, but because they come from the same ancestral origin. The forelimbs of a human, a bird, a whale, and a dog may seem to serve wildly different roles, grasping, flying, swimming, running. But underneath it all, the bones are arranged in much the same pattern. It's like seeing variations on a theme, shaped by millions of years of adaptation. Sometimes, nature leaves behind structures that seem to have no current function at all, the evolutionary leftovers. These are vestigial structures. Think of the wings on an ostrich, which can't lift it into the air, or the tiny internal leg bones found in modern whales, long removed from the limbs their ancestors once walked on. Even cacti, though adapted for desert life, still sprout tiny leaves, a reminder of a time when their ancestors had plenty of water to spare. Then there's convergent evolution, a fascinating reminder that evolution can arrive at the same solution more than once. Unrelated species, living in similar environments, often end up with similar traits, not because they're cousins on the tree of life, but because they've faced similar challenges. The Arctic fox, for example, and some birds of snowy landscapes both develop white coats in winter. It's not a sign of shared ancestry, just shared pressures. A world where blending into the snow means the difference between survival and becoming someone's lunch. Even the study of embryos, embryology, gives us clues about evolution's reach. During early development, the embryos of animals as different as fish, birds, and humans can look strikingly alike. Gill slits, tails, and other features appear, only to fade or transform as development continues. These similarities aren't coincidences. They're echoes of a common origin, glimpses of a time when these species were much more alike than they are now. The way living things are spread across the planet tells a fascinating evolutionary story, one that makes much more sense when we factor in the slow drift of continents over deep time. Life isn't randomly scattered. Its distribution reflects history, geography, and the long, winding paths evolution has taken. Some groups of organisms can be found across many continents, and this usually points to a very ancient origin, one that predates the breakup of the supercontinent Pangaea. These creatures were around when the landmasses were still connected, and they simply traveled with the continents as they slowly drifted apart. Other groups, though, are much more local. Australia, for instance, has a whole cast of characters you won't find anywhere else: kangaroos, koalas, wombats, and other marsupials. That's because Australia split off early and evolved in isolation. While the rest of the world was going in one evolutionary direction, Australia's wildlife took its own route, resulting in a wonderfully odd and unique menagerie. Isolated islands offer even more vivid examples. When a group of organisms becomes separated from its relatives, say on a remote island, evolution can take unexpected turns. The Galapagos Islands are a classic case, with finches that diverged into entirely new species, each adapted to a slightly different ecological niche. Hawaii, too, has its own parade of species found nowhere else on Earth. These island creatures often bear traces of their mainland ancestry, but they've become something new, a quiet demonstration of evolution at work. And then, if we zoom in, way in, to the molecular level, we find some of the strongest evidence of all. Every living thing uses DNA as its genetic material. And not just that. The way DNA is copied, read, and translated into proteins is nearly identical across all known life. This remarkable consistency points to a shared origin, a single thread that runs through the entire living world. Of course, DNA isn't identical in every species. There are differences, and these differences are exactly what we'd expect to see if life had diversified gradually over time. Closely related organisms share more of their DNA. More distantly related ones, less. It's like looking at old family photo albums. You can trace the relationships, the similarities, and the branching paths. DNA has also given us insight into how new traits come about. Sometimes a gene will accidentally make a copy of itself. One copy continues doing its usual job, while the other is free to mutate without causing harm. Over time, that second copy might develop an entirely new function, a new protein, a new adaptation, a new twist in the story. Taken together, the evidence is overwhelming. From fossils in ancient rock, to bone structures and embryonic development, from island species to the deep language of DNA, it all points in the same direction. Life on Earth has changed, is still changing, and will continue to change, shaped by the slow, persistent force of evolution. When we talk about evolution, we often focus on natural selection, the quiet, relentless force of nature shaping life over time. But there's another player in this story, one that's relatively new and remarkably influential: us. Humans have had a significant hand in shaping the traits of many species, especially those we find useful, tasty, or simply pleasant to have around. This process is known as artificial selection, or selective breeding. And it works in much the same way as natural selection. Only instead of nature doing the selecting, we do. By choosing which individuals get to reproduce, humans have guided the development of everything from crunchy apples to woolly sheep. Over generations, these choices build up, sculpting entire breeds of animals and varieties of plants. When it's animals, we usually call them breeds. Think golden retrievers or dairy cows. In plants, you'll hear terms like cultivars, varieties, or hybrids. And when two different breeds or varieties are crossed, we get crossbreeds or hybrids, depending on whether it's a sheep or a squash. There are a couple of common approaches to artificial selection. One is fairly straightforward. Pick the best individuals, the tallest wheat, the fastest horse, the chicken that lays the most eggs, and allow only those to reproduce. Do this for a few generations, and the trait becomes more common. It's the traditional breeder's method: select, repeat, refine. Another approach is a bit more hands-off, sometimes called controlled natural selection. In this method, breeders don't pick the individuals directly. Instead, they create a specific environment and let nature take its course, observing which individuals thrive under those conditions. It's still artificial selection, but with a looser grip on the wheel. There's also a third route, mostly used in research: selection experiments. These are designed to measure how quickly and how strongly certain traits can evolve under selective pressure. They're usually more about observation than agriculture, but they've helped us understand just how powerful selection, whether natural or artificial, can be. In animal breeding, various techniques are used to maintain or amplify desired traits. Inbreeding and line breeding focus on keeping genetic lines tight, while outcrossing brings in unrelated individuals to add variation or counteract undesirable traits. Plant breeders use similar tools, sometimes with seeds, sometimes with cuttings, sometimes with a touch of pollen and a bit of patience. The results are all around us: tomatoes bred for sweetness, sheep bred for softer wool, corn with kernels that grow in perfect rows. Much of what fills our kitchens and gardens is the result of centuries, and sometimes millennia, of artificial selection. Historically, this practice didn't just feed civilizations, it also helped scientists understand how evolution works. When people saw that careful breeding could transform animals and plants over just a few generations, it became clear that selection, even without conscious intention, was a powerful force. And not all artificial selection is deliberate. Sometimes our actions unintentionally shape the traits of species. A farming practice might favor larger seeds, not because anyone selected them, but because they happen to fall into better soil or be easier to harvest. Over time, traits like that stick around. Evolution doesn't care if the pressure is deliberate. It just responds. In many cases, domestication has been a blend of the planned and the accidental. Human hands guided some traits, while environmental conditions, farming methods, and chance shaped others. Together, they've given us the rich diversity of domesticated life we rely on today. Speciation is the process by which new species come into being, the branching points in the great tree of life. Even though all living things on Earth share some deep genetic common ground, not every group of organisms can mix their genetic material and produce offspring. In biology, the rule of thumb is this: if two organisms can reproduce together, and their offspring can also go on to reproduce, they're considered part of the same species. It's a practical definition, one focused on the continuity of life through generations. Of course, things are rarely as tidy in nature as definitions might suggest. But generally speaking, when two populations stop being able to interbreed, either because of genetic differences, behavioral changes, or physical separation, they start drifting down their own evolutionary paths. Over time, that drift can become a divide, and that divide can eventually mark the emergence of a brand new species. A widely accepted definition of a species is quite straightforward. It's a group of individual organisms that can interbreed and produce fertile, viable offspring. If two organisms can do that, and their offspring can do it too, then they're considered part of the same species. If they can't, at least not reliably or naturally, they're probably from different species. This definition works especially well for eukaryotes, the more familiar branches of life, like plants, animals, and fungi. Things get a little more complicated when we talk about bacteria and other prokaryotes, which don't reproduce in the same way. But for now, we'll keep our focus on the organisms where this definition fits best. Members of the same species tend to look and function in similar ways. These shared traits, both inside and out, all come down to DNA, the set of instructions each organism carries. The closer the genetic relationship, the more DNA they share. Much like a family tree, your DNA is more similar to your parents than to a distant cousin's. In the same way, individuals within a species have the highest level of genetic similarity, which allows them to produce fertile offspring together. That said, appearances can be deceiving. Some members of the same species might look quite different from one another, and yet they're genetically close enough to reproduce. Domestic dogs are a good example. A Great Dane and a Chihuahua might look like they're from different planets, but they're still the same species, capable of producing puppies, if not without a few logistical challenges. On the flip side, some organisms can look very much alike, but actually belong to different species. Consider the bald eagle and the African fish eagle, two impressive birds with similar features and behaviors, but genetically, they're far enough apart that even if someone were to engineer a hybrid, the resulting offspring would almost certainly be infertile. Their genetic instructions just don't line up well enough to continue the line. Within a species, all individuals share what's called a gene pool, a collection of all the genetic variation available in that population. And for evolution to happen, it's the gene pool that must change. Any lasting change in a population has to be genetic. Only genetic changes can be passed on to future generations. There are two ways this happens, depending on how reproduction works. In asexual reproduction, a genetic change in one cell can be passed directly to its offspring. It's a copy and paste situation. But in sexual reproduction, the process is a bit more selective. Only changes that occur in the reproductive cells, the eggs and the sperm, can be inherited. A mutation in your skin or your liver won't reach your children, but a change in a gamete just might. So for evolution to take hold, for a new trait to spread and become part of a species, reproduction is essential. Without it, variation goes nowhere. But with it, even the smallest genetic change has the potential to echo far into the future. When biologists talk about a species, especially in the context of sexually reproducing organisms, they usually mean a group of individuals that can actually, or at least potentially, interbreed and produce fertile offspring. It's a neat definition, and for the most part, it works. But as always in biology, there are exceptions. Sometimes species that are closely related can produce hybrid offspring, a kind of genetic handshake across species lines. These hybrids don't always thrive or reproduce, but their very existence tells us something important. The line between species isn't always sharp. In fact, when hybrids show up in nature, it often hints that the species involved may have only recently diverged, and that the process of separation might still be underway. Given the incredible variety of life on Earth, there must be powerful forces behind the formation of new species, a process we call speciation. Darwin imagined speciation as a branching pattern, one ancestral species splitting into two, then four, then many, like the branches of a tree spreading out as time moves forward. Some branches flourish, others fade, and new ones are always forming, as long as the conditions are right. For speciation to occur, one population must split into two or more groups that can no longer interbreed. Over time, they evolve independently, and the genetic distance between them grows. Eventually, even if they were brought back together, they might not recognize each other as potential mates, or their offspring might not be viable. At that point, we consider them distinct species. Scientists group the mechanisms of speciation into two broad categories: allopatric and sympatric speciation. Allopatric speciation, meaning different homeland, is the more common route. It happens when populations are physically separated by mountains, rivers, oceans, or simply distance. Once separated, gene flow between them stops, and evolution begins to take its separate courses. Sympatric speciation, on the other hand, means same homeland. This is where species diverge even while living in the same place. A trickier path, but not impossible. It often involves behavioral changes, timing shifts in reproduction, or ecological preferences that slowly pull groups apart without a physical barrier. Although we usually think of speciation as one species splitting into two, more than two species can sometimes emerge, especially in cases of rapid diversification. Still, it's easiest to imagine it as a series of splits over time, gradual, quiet, and cumulative. Now, in a continuous landscape where individuals can move freely and mate with others across the range, gene flow tends to keep everyone genetically similar. But once a population is separated, say by a new river or a patch of desert, that flow stops. From that point on, each group experiences its own mutations, its own selective pressures. Different climates, predators, and resources start shaping each population in unique ways. This physical separation can happen through dispersal or vicariance. Dispersal is when a small group moves away to a new area. Maybe birds fly to a new island, or seeds drift across the sea. Vicariance is when a once-connected population gets split by a geographic change. A mountain rises, a glacier advances, a forest becomes a plain. In both cases, once the populations are isolated, evolution begins to work on them independently. There are many well-documented cases of allopatric speciation. Take the spotted owls along the west coast of North America. In the north, you'll find the northern spotted owl. In the south, the Mexican spotted owl. These groups have been shaped by the different environments they live in. Cool forests versus warmer, drier landscapes. Over time, they've developed different hunting habits, slight differences in appearance, and most importantly, genetic differences that mark their evolutionary divergence. In general, the farther apart two populations are, and the longer they're apart, the more likely they are to evolve into distinct species. Distance brings different challenges, and evolution responds accordingly. Sometimes speciation happens not just because of separation, but because a species spreads out and adapts to different ecological roles. This is called adaptive radiation. A single species fans out into many, each one specialized for a particular niche. It's a common pattern on island chains, where geographic isolation is built right into the landscape. The Hawaiian Islands are a textbook example. One ancestral species arrives, and before long, you have a burst of new species, each tailored to its own little corner of the environment. The Hawaiian honey creepers are a beautiful case of this. From a single ancestral bird, an entire flock of species evolved, some with long, curved beaks for nectar, others with thick, strong beaks for cracking seeds. Each species took its own evolutionary path, shaped by the food it found, the place it settled, and the challenges it faced. Darwin's finches in the Galapagos Islands follow the same pattern. One ancestor, many outcomes, each one a variation shaped by the slow, steady work of natural selection. So what about cases where there's no physical barrier, no ocean, no mountain range, no canyon cutting a population in two? Is it still possible for species to split when they're living side by side? The answer is yes, and the process is known as sympatric speciation. As the name suggests, sympatric means same homeland. In this case, new species arise not by being physically separated, but while sharing the same space. It's less common than geographic separation, but it does happen, and scientists are still uncovering exactly how. One route sympatric speciation can take begins not with geography, but with chromosomes, specifically when something goes wrong during cell division. Usually cells split their chromosomes neatly between the new cells, keeping the number balanced. But sometimes the split isn't so tidy. The result can be a cell with too many or too few chromosomes, a condition known as aneuploidy. It's one possible source of reproductive isolation. Then there's a more dramatic version, polyploidy. This occurs when a cell ends up with entire extra sets of chromosomes. In other words, instead of 2 sets, one from each parent, the cell might end up with 3 or 4. This happens most often in plants, and surprisingly, it can create new species almost instantly. There are 2 main kinds of polyploidy that can lead to speciation. The first is called autopolyploidy. Auto meaning self. Here, all the extra chromosome sets come from the same species. It usually starts with a mishap during the formation of gametes, egg or sperm cells, where instead of splitting neatly, all the chromosomes end up in one gamete. Imagine a plant species where the usual number of chromosomes is 6. A normal gamete would have 3, but if something goes wrong, the gamete might keep all 6. If 2 such autopolyploid individuals reproduce, their offspring will have 4 full sets of chromosomes, making them genetically incompatible with the original population. And just like that, a new species may form. The second kind is called allopolyploidy. Allo meaning other. This happens when 2 different species interbreed and their chromosomes combine. At first, the resulting hybrid may be sterile, but if chromosome doubling occurs, and sometimes it does, the hybrid can suddenly become fertile. It now carries a full, balanced set of chromosomes, allowing it to reproduce, but only with others like itself. Many of our cultivated crops, wheat, cotton, and tobacco among them, owe their existence to this kind of speciation. While polyploidy is relatively rare in animals, in the plant world, it's surprisingly common. In fact, more than half of all plant species studied seem to have polyploidy somewhere in their evolutionary history. Some scientists even suggest that polyploidy isn't just a genetic hiccup. It might be an evolutionary strategy, helping plants quickly adapt to changing environments. Now, whether speciation begins with chromosome changes, geographic separation, or anything else, it usually ends in the same place: reproductive isolation. Over time, genetic changes start to affect how and whether individuals can successfully mate and reproduce. Even if members of 2 diverging groups were to meet again, mating might be difficult, or simply unlikely. And if mating does happen, the offspring might not survive, or might be sterile. Biologists group these barriers to reproduction into 2 broad types: prezygotic and postzygotic. Prezygotic barriers happen before fertilization. They prevent mating altogether or block the fertilization process. Postzygotic barriers come into play afterward, affecting the survival or fertility of the resulting offspring. When two gametes, an egg and a sperm, merge, the result is a zygote. It's the very first cell of a new organism. But getting to that point isn't always easy. In fact, much of the time, it doesn't happen at all. That's where reproductive barriers come in. These are the obstacles that prevent different species from successfully mating and producing fertile offspring. We group these barriers into two main categories: prezygotic and postzygotic. Prezygotic barriers stop reproduction before fertilization ever occurs. Postzygotic barriers come into play after fertilization, for instance, if the embryo doesn't survive, or if the resulting offspring is sterile. Prezygotic barriers can take many forms. One common type is temporal isolation. Some species breed only at certain times of the year, or even at specific times of day. If two closely related species don't overlap in their mating seasons, they simply won't have the opportunity to reproduce. For example, two frog species might share the same habitat, but have different breeding windows. One starts in January, the other in March. Despite living side by side, they remain reproductively separate. Habitat isolation works in a similar way. If a population moves to a new environment, even a nearby one, and stops overlapping with its relatives, reproduction with the original group may cease. Over time, genetic differences can build up, and speciation may quietly unfold. Picture a population of crickets separated by a flood, each group adapting to a different patch of land. One drier, one more shaded, each selecting for different traits. Then there's behavioral isolation. Many animals rely on courtship rituals to identify suitable mates, and these behaviors are often species-specific. A classic example is fireflies. Male fireflies use unique light patterns to attract females. If a male sends out the wrong signal, the female doesn't respond. No response, no mating. Behavioral mismatches can be subtle but extremely effective barriers. Some barriers are physical. Gametic isolation occurs when sperm and egg cells from different species simply can't fuse. Even if mating happens, fertilization doesn't. In other cases, mechanical incompatibility prevents mating from succeeding at all. For example, male damselflies of different species have uniquely shaped reproductive organs. A poor anatomical fit means that, despite effort, reproduction is off the table. Plants have their own mechanical barriers. Some flowers are shaped in ways that attract only specific pollinators. A nectar tunnel might be perfectly suited to one species' tongue or beak, but too long or too narrow for another. This, too, keeps species from crossing genetic lines. Even when fertilization does occur, postzygotic barriers may still prevent successful reproduction. One outcome is hybrid inviability, where the embryo fails to develop properly and dies early. Another is hybrid sterility. The hybrid is born, grows up, but can't reproduce. The classic example is the mule, a hybrid of a horse and a donkey, strong and healthy, but sterile. Polyploidy, as we've discussed, is one well-known path to sympatric speciation, especially in plants. But it's not the only way species can diverge without a physical barrier. Imagine a large lake filled with a single species of fish. As the population grows, so does competition for food. Some individuals might begin feeding at different depths, perhaps closer to the lake bed or near the surface. Over time, These fish start to associate more with others in the same feeding zone. Eventually, they begin mating primarily within that group. The deeper dwellers evolve differently from those near the surface. Given enough time and enough isolation in behavior and diet, the two groups may become separate species, even though they still share the same lake. This kind of sympatric speciation isn't just theoretical. It's been observed. One striking example comes from Lake Victoria in Africa, where cichlid fish have diversified into hundreds of species, often in relatively short evolutionary timeframes. Similar patterns have emerged in other lakes, such as those in Nicaragua, where cichlid populations in the same body of water have adapted to different ecological niches, feeding on different prey, occupying different zones, and eventually developing distinct forms. Even after two populations have diverged and become distinct species, their evolutionary story might not be entirely separate. In the natural world, organisms don't usually stop and ask whether they belong to the same species. If two individuals are biologically capable of mating and happen to be in the same place at the same time, they often will. When two closely related species come back into contact and begin to interbreed, they can form hybrid offspring. This creates what biologists call a hybrid zone, a region where the boundaries between species begin to blur. In these zones, individuals from both groups continue to interact, and their offspring carry a mix of traits from each parent species. What happens next depends on a few key factors, especially how successful those hybrids are, and whether reproductive barriers are still in place. In some cases, the hybrids are less fit than either of the parent species. They may have lower survival rates or struggle to reproduce. When this happens, a process called Moir reinforcement may occur. Natural selection begins to favor individuals who don't mate with members of the other species, simply because their offspring don't do as well. Over time, this strengthens the reproductive barriers between the two groups, nudging them even further apart. It's nature's way of closing the door on the hybrid option. But sometimes, the opposite happens. If hybrids are just as fit, or perhaps even better suited to the environment, the reproductive barriers may begin to break down. Gene flow between the two groups increases, and over generations, the distinctions between them fade. This can lead to fusion, where two species effectively merge back into one. The separation that once marked them as distinct starts to dissolve. There's also a third possibility, a kind of middle ground. In some cases, hybrids are roughly as fit as their parents, and the barriers between the species remain, but they don't grow stronger or weaker. The hybrid zone settles into a kind of equilibrium. Hybrids continue to be produced in a specific area, while the parent species persist elsewhere. This situation, known as stability, can last for a long time, a gentle balancing act between divergence and blending. Hybrid zones offer a fascinating glimpse into the ongoing nature of evolution. They show us that speciation isn't always clean and final. Sometimes the process runs in reverse. Sometimes it pauses. Scientists all over the world study speciation, not only by observing living organisms, but also by examining the fossil record, where ancient life has left behind its quiet imprint in stone. As more evidence is uncovered, researchers refine their understanding of how life evolves, developing models to explain the pace at which new species emerge. There are two main patterns that scientists use to describe how speciation might unfold over time. the gradual speciation model, and the punctuated equilibrium model. In the gradual model, change happens slowly, bit by bit. Small genetic variations build up over generations, and over long stretches of time, these subtle shifts accumulate. Eventually, the population drifts far enough from its origins that it becomes a new species. It's a smooth, continuous process. Evolution by slow erosion and steady shaping. The punctuated equilibrium model, by contrast, paints a different picture. In this model, speciation happens in quick bursts, geologically speaking at least. A new species may appear rapidly, branching off from the parent species in response to sudden environmental changes or isolation. After this initial burst of change, the punctuation, the new species may remain largely unchanged for long periods. This creates a pattern of quick divergence, followed by long-term stability or equilibrium. It's worth noting that these two models aren't mutually exclusive. Nature isn't bound to choose one tempo over another. Some species evolve slowly and steadily. Others appear suddenly in the fossil record, hinting at a more abrupt transition. The pace of speciation often depends on external factors, especially environmental conditions. When environments change dramatically or rapidly, evolution may accelerate. A species that once seemed stable can suddenly find itself facing new pressures. And if a portion of the population becomes isolated, the stage is set for change. Imagine a species of snail that's remained more or less the same for thousands of years. Its fossil record would look remarkably consistent. But then, one day, the water level in its lake drops. A small group of snails finds itself cut off from the rest, now living in a slightly different habitat, perhaps with new predators, less food, or different conditions altogether. This smaller population carries only a fraction of the genetic variation found in the original group, and because it faces new challenges, traits that happen to help with survival can quickly become dominant. In this scenario, evolution may move faster, not because nature is in a hurry, but because the pressures are suddenly stronger, and the population is smaller, more responsive to change. All life on Earth is connected. That's one of the most powerful ideas to come out of evolutionary theory. Whether we're talking about humans, beetles, trees, or bacteria, every organism alive today can trace its lineage back to a common ancestor, a simple life form from the distant past. Over millions of years, evolution has shaped each of these lineages into the distinct forms we see today. Some changes have been slow and steady, others sudden and dramatic, but all are part of the same unfolding story, one shaped by time, chance, and the relentless pressure of the environment. At the heart of this process is natural selection, the quiet force that favors traits and behaviors that help an organism survive and reproduce. Traits that work well tend to stick around. Those that don't tend to fade away. But natural selection isn't a creator, it's a filter. It can't invent new traits out of thin air. It can only work with what's already there. So where do new traits come from? That role belongs to mutation. Mutations are small changes in an organism's genetic code. Most are neutral, some are harmful, but every once in a while, a mutation gives an individual a slight advantage, a better beak, a stronger shell, a slightly sharper eye. That new trait, once introduced, can spread through the population over time, especially if it helps with survival or reproduction. Along with other sources of variation, mutations provide the raw material that evolution works with. Combined with natural selection and other forces like genetic drift, gene flow, and environmental pressures, they drive the vast diversity of life on this planet. Genes don't always read from the same script. Many come in alternative versions called alleles. And those variations are what give traits their subtle, and sometimes not so subtle, differences. Each individual carries 2 alleles for a given gene, but a population can hold many more. Think of the ABO blood group. 3 possible alleles help determine whether your red blood cells advertise A, B, both, or neither. When Gregor Mendel was busy counting pea plants, he unwittingly opened the door to a new field. By the early 20th century, biologists began asking a bigger question. How do allele frequencies shift in entire populations over time? That line of inquiry became population genetics. In this context, allele frequency is simply the proportion of a particular allele in the population's gene pool. Evolution, at its core, is just a change in those frequencies from one generation to the next. If the proportion of the IA allele in the ABO system rises or falls, evolution has happened, quietly, mathematically, and often invisibly. Why do these frequencies budge? One obvious culprit is natural selection. When a certain allele gives its carriers a survival or reproductive edge, they tend to leave more offspring. Those offspring inherit the winning ticket, and over time the advantageous allele can spread through the population, sometimes all the way to lung fixation, where everyone carries it. Harmful alleles, by contrast, are usually shown the exit, but nature isn't always so purposeful. Allele frequencies also drift around thanks to sheer chance, a process called genetic drift. Picture flipping a coin only 10 times. You won't always get that perfect 50-50 split of heads and tails. In small populations, the gene pool can wobble the same way, letting some alleles rise or vanish without any adaptive rhyme or reason. A special case of drift is the founder effect. Imagine a handful of individuals striking out to colonize a new patch of habitat. The genetic snapshot they carry may look very different from the larger population they left behind, simply because of who happened to make the trip. Over generations, that random sample can blossom into a population with a distinct genetic identity. In real life, natural selection and genetic drift often work simultaneously, nudging and tugging allele frequencies in their own quiet tug-of-war. Separating their effects can be tricky, but together they explain how gene pools shift, diversify, and sometimes march to the beat of pure chance. In any given population, individuals often show different phenotypes. That is, they express different physical traits or behaviors. These differences usually reflect the underlying variation in their genes. When 2 or more variations of a specific trait exist within a population, we call it polymorphism. Populations like these are said to be polymorphic, and they're essential to evolution. The distribution of these traits, who has what and how common each variation is known as population variation. It's influenced by both the genetic makeup of the population and the surrounding environment. And understanding what drives this variation is key to predicting how a population might change over time, especially under evolutionary pressure. Natural selection and other evolutionary forces primarily act on traits that are heritable, traits encoded in the DNA and passed from parents to offspring. Alleles that give an organism an edge, whether through sharper eyesight or a useful behavior, tend to become more common because they increase survival, or reproductive success. On the other hand, alleles that cause harm are usually selected against and become rarer over time. It's important to note that not all traits are inherited. Some are acquired, shaped by lifestyle, effort, or environment. For example, an athlete might build strong muscles through training, but those muscles won't be passed on to their children. However, if there's a genetic basis for strength or endurance, that potential can be inherited. The degree to which a trait is shaped by genes rather than the environment is known as heritability. The higher the heritability of a trait, the more responsive it is to evolutionary forces. Natural selection can only work on what's passed down, so heritable variation is evolution's raw material. The total genetic variation within a population, what we call genetic variance, helps maintain phenotypic diversity. It's also crucial for long-term survival. That's why conservation efforts often focus on preserving genetic diversity. Without it, populations are more vulnerable to inbreeding. Inbreeding happens when closely related individuals reproduce. In large, diverse populations, rare harmful alleles tend to remain hidden because it's unlikely that two carriers will meet and mate. But in small or inbred populations, those chances rise. The result can be the increased expression of harmful recessive traits, genetic disorders, lower fertility, or reduced overall fitness. Now, when we look at how allele frequencies change in a population, we begin to see the dynamics of evolution in motion. Natural selection is one of the primary drivers. Take gorillas, for instance. A large, dominant male, a silverback, might win more mating opportunities. His traits, including body size, become more common as his offspring spread those genes. Over time, natural selection favors certain physical traits or behaviors, and the gene pool shifts accordingly. But not all changes in allele frequency are driven by selection. Sometimes, it's simply chance. This is the domain of genetic drift. Random fluctuations in allele frequencies, especially in small populations. If one individual in a group of 10 dies without reproducing, that's 10% of the population's genetic material gone in a single moment. No selection, no pressure, just randomness. In larger populations, this kind of drift has less impact, but in small groups, it can dramatically reshape the gene pool. A more extreme form of genetic drift is the bottleneck effect. This happens when a sudden event, a flood, fire, disease, or other disaster, wipes out a large portion of the population. Only a few individuals survive, and the next generation reflects only their genetic material. Much of the original diversity is lost in the blink of an evolutionary eye. Another form of drift is the founder effect. Imagine a small group breaking off from a larger population and settling in a new environment. The genes they carry may not represent the full spectrum of the original group. Over time, their descendants develop a new genetic identity, not through adaptation necessarily, but because of the limited sample that made the journey. Another key player in the evolutionary story is gene flow. The movement of alleles into or out of a population through migration. As individuals travel, or as gametes like pollen drift from one area to another, new genetic material can be introduced into a population. This process changes allele frequencies and adds to the population's genetic diversity. Gene flow is especially common in species with wide geographic ranges, plants that rely on wind or insect pollination, birds that migrate between habitats, and fish that move through connected waterways all can carry genes from one group to another. When gene flow is active, populations stay more genetically similar. When it's limited, they can start to diverge. Mutation is another force behind evolutionary change. Mutations are changes in an organism's DNA, often random, sometimes silent, but always significant over time. Most mutations are neutral, causing no noticeable effect. Some are harmful and tend to be removed by natural selection. But now and then, a mutation provides a useful advantage, a slightly stronger bone, a more efficient enzyme, and that tiny change can ripple through a population if it boosts survival or reproduction. Over long timescales, the accumulation of these genetic changes fuels the diversity of life. Non-random mating is yet another influence on a population's genetic makeup. In many species, individuals don't choose mates at random. They often prefer partners with certain traits, a behavior known as assortative mating. This can reinforce specific phenotypes within a group. For example, birds may choose mates based on song, color, or size. Over time, these preferences can shape the genetic structure of a population. Geographic separation also plays a role. Physical barriers like mountains, rivers, or even urban development can prevent individuals from mating, gradually isolating populations. When these groups remain separated long enough, their traits may start to diverge, sometimes slowly, sometimes sharply. Environmental factors influence phenotypes too. For instance, human populations living in regions with high sunlight exposure often develop darker skin, an adaptive response to ultraviolet radiation. Some reptiles and fish exhibit temperature-dependent sex determination, where the ambient temperature during development influences whether the embryo becomes male or female. It's a fascinating reminder that environment and genetics are always in conversation. Differences in phenotype across geographic regions are known as geographic variation. These differences can be subtle, like the gradual increase in body size among animals living in colder climates, a trend explained by the need to conserve body heat. Or they can be abrupt, like flowering plants blooming at different times depending on elevation, a pattern known as an altitudinal cline. When gene flow occurs between these geographically separated populations, the result can be a smooth gradient of traits, a gradual change across space. But when gene flow is restricted, the differences can deepen. Over time, this divergence may become so pronounced that it leads to the formation of new species. Natural selection acts not on isolated genes, but on entire organisms, shaping evolution through the success or failure of individuals to survive and reproduce. Within a population, individuals carry a mix of beneficial and harmful alleles. If a helpful trait is paired with a lethal one, say a gene that improves reproductive success alongside one that causes a fatal disease in early childhood, then that individual won't pass on either trait. They won't survive long enough. This is why natural selection operates at the level of the individual, not the gene, and why we describe success in terms of evolutionary fitness, a measure of how much an individual contributes to the next generation's gene pool. Fitness isn't about strength or speed in absolute terms. It's always relative. What matters is whether an individual contributes more offspring than others in the population. This comparative measure is known as relative fitness, and it helps researchers understand how populations shift and evolve over time. Natural selection can influence population variation in a number of ways, including stabilizing selection, directional selection, diversifying selection, frequency-dependent selection, and asexual selection. Each of these modes affects which traits become more common and how much genetic variation is preserved or lost in the process. In a stabilizing selection, natural selection favors the average, selecting against extremes. Consider a population of mice in a forest. If the soil is brown, then mice with fur that's too light or too dark may stand out to predators, while those with medium brown coats blend in best. Over generations, the population becomes more uniform in color, and genetic variance narrows. By contrast, directional selection occurs when the environment changes in a way that favors one extreme over the others. A famous example is the peppered moth in England. Before industrial pollution, light-colored moths were camouflaged against lichen-covered trees. But as soot darkened the landscape, darker moths had the advantage. Their numbers rose, and the population's coloration shifted, a textbook case of environmental change driving evolutionary direction. Then there's diversifying selection, also known as disruptive selection. This happens when individuals at both extremes of a trait have an advantage, while those in the middle are less fit. In some species, large males may dominate through strength, while small males sneak around and successfully mate by deception. Medium-sized males caught between strategies are often left behind. Similarly, in habitats with patchy backgrounds, say light sand and dark vegetation, light and dark mice may each find camouflage, while medium-colored mice stand out everywhere. This type of selection increases genetic diversity, sometimes dramatically. Frequency-dependent selection adds another twist. Here, the success of a trait depends on how common or rare it is. In positive frequency-dependent selection, common traits are favored. The more common something is, the better it works. But in negative frequency-dependent selection, rarity is the advantage. A great example comes from side-blotched lizards in the Pacific Northwest. These lizards come in 3 throat colors, each linked to a different reproductive strategy. Orange males are aggressive and territorial, blue males are cooperative and pair bonding, and yellow males mimic females to sneak in unnoticed. Their success depends on what strategies are most common around them. When orange males are common, yellow males do well. When yellow males are common, blue males succeed. And so the cycle continues. This dynamic keeps genetic diversity high and ever shifting. Lastly, we arrive at sexual selection, a powerful force especially in animals. In many species, males and females differ not just in reproductive organs, but in size, color, and behavior, traits known as sexual dimorphisms. Males are often larger, more colorful, or more ornamented. Think of a peacock's tail or the elaborate dances of tropical birds. These traits don't help them hide or survive predators, but they do attract mates. Why such showiness? Because in many species, male reproductive success is highly variable. Some males may mate frequently, while others don't mate at all. So any trait that gives a male an edge, whether it's physical dominance or decorative flair, gets passed on. Over generations, sexual selection exaggerates those traits, even if they come with trade-offs like increased visibility or energy costs. Females, unlike males, often have fewer mating opportunities. As a result, they tend to be more selective, choosing mates carefully. often preferring the most desirable males. This preference plays a central role in sexual selection, a process that can produce traits not necessarily helpful for survival, but useful for securing mates. Sexual dimorphism, the physical differences between males and females, varies widely across species. In many, males are larger, louder, or more ornamented, but in others, the roles are reversed. Some species have females competing for mates, with males doing the choosing. In these cases, it's the females that show greater variation in reproductive success, and selection favors traits like larger size and striking appearance, traits we often associate with males in other species. Sexual selection can produce what are called secondary sexual characteristics, traits that improve reproductive success, even if they come at a cost to survival. The peacock's tail is a classic example. It's extravagant, beautiful, and utterly impractical. It slows the male down and makes him more visible to predators. But the very fact that a male can survive with such a burden might be the point. This idea is known as the handicap principle, the notion that only the fittest individuals can afford the cost of an exaggerated trait, and so the trait itself becomes an honest signal of genetic quality. Another explanation, closely related, is the good genes hypothesis. This suggests that elaborate ornaments signal a male's underlying genetic health, perhaps his resistance to disease or metabolic efficiency. Females who choose these showy males might produce fewer offspring, but those offspring may inherit genes that help them survive and reproduce. In evolutionary terms, fewer but fitter can be a winning strategy. Both the handicap principle and the good genes hypothesis suggest that these flashy traits are more than decoration. They are honest signals of the male's quality, giving females a way to select mates who will pass on strong, resilient genes to the next generation. While sexual selection and natural selection can drive impressive evolutionary changes, it's important to remember that natural selection has its limits. It can only act on what already exists, traits shaped by mutation, gene flow, and the shuffling of alleles. It doesn't create new solutions from scratch. It simply chooses among the available options. Natural selection also acts on individuals, not on individual alleles. And because many alleles are inherited together, linked on the same stretch of DNA, a beneficial trait might be paired with a harmful one. If the combination still leads to high overall fitness, it can persist. But this also means that some advantageous alleles may be lost if they're consistently paired with disadvantageous ones. The result is a balancing act, with fitness depending on the whole organism, not just one gene at a time. Sometimes even beneficial changes can't take hold because of what lies in between. Evolution doesn't always have a clear path forward. Imagine a population of light-colored mice that blend with sandy soil and a dark variant that blends with dark grass. The dark mice might be better suited to a changing environment, but shifting from light to dark requires passing through intermediate coat colors that don't camouflage well with either background. Those intermediates are vulnerable, and so the population may get stuck in place, not because the destination isn't better, but because the journey carries too much risk. And finally, not all evolution is adaptive. While natural selection favors traits that increase fitness, other forces, like genetic drift and gene flow, can introduce harmful traits or neutral ones that persist for generations. Evolution isn't trying to create perfect organisms. It has no direction, no end goal. It's simply the outcome of competing pressures, random events, and the endless reshuffling of genetic material. Let's talk about one of the foundational ideas in population genetics, the sucker for Hardy-Weinberg principle of equilibrium. First introduced in the early 20th century by English mathematician Godfrey Hardy and German physician Wilhelm Weinberg, this principle describes how we'd expect genes to behave in a population where no evolutionary forces are acting. It's a bit like imagining a perfectly calm pond. No ripples, no wind, no fish darting beneath the surface. Just a still body of water, undisturbed. According to the Hardy-Weinberg principle, the frequencies of alleles and genotypes in a population will remain constant from one generation to the next, as long as certain conditions are met. If nothing disrupts the system, no mutations, no migration, no natural selection, and no random changes due to small population size, then the genetic makeup of that population will stay the same. To be precise, for the Hardy-Weinberg equilibrium to hold true, a few key assumptions must be met. The population must be infinitely large. Individuals must mate randomly. There must be no mutations altering genes. No one can leave or enter the population, and no trait can give any reproductive advantage over another. Of course, in the real world, these conditions are never fully met. No population is truly infinite. Mating isn't always random. Mutations happen, organisms migrate, and natural selection is almost always at work. But that doesn't make the Hardy-Weinberg principle any less important. In fact, it's a vital tool, a theoretical baseline we can use to compare real populations and understand when, how, and why they're evolving. In population genetics, different alleles are represented mathematically, often as variables like P and Q. From these, scientists can estimate not just how common an allele is, but how often different combinations of those alleles, or genotypes, appear in the population. This overall pattern of genotype distribution is known as the genetic structure of the population. That structure is important because it shapes the phenotypes we can observe, the physical traits we see in the living world, but it's not always straightforward. For many traits, especially those controlled by dominant and recessive alleles, only the homozygous recessive genotype reveals itself clearly. For instance, if a particular trait only shows up when an individual carries 2 copies of a recessive allele, we can count how often that trait appears, and then use the Hardy-Weinberg equations to estimate how frequent the other genotypes must be. If a population were truly at equilibrium, untouched by selection, mutation, migration, or drift, then its allele frequencies would remain unchanged over time. The Hardy-Weinberg equation would hold generation after generation, but Hardy and Weinberg themselves understood that this is an ideal, not a reflection of real life. Nature is not still water. It's a river, always moving. And that's where the Hardy-Weinberg model becomes so powerful. It gives scientists a way to measure change. When a population doesn't follow the expected pattern, it's a signal that something is at work. Perhaps selection, perhaps drift, perhaps a wave of migration, introducing new genetic material. By comparing what we see in real populations with the Hardy-Weinberg baseline, we can begin to understand how evolution is acting. A bee and an echinacea flower may not look like they have much in common, but like all living things, they share a long, tangled history. By tracing patterns in anatomy, behavior, and DNA, scientists piece together how life has changed, and how it's connected. From single-celled beginnings to everything that walks, swims, flies, or blooms today, evolution helps us understand how life keeps branching, adapting, and surviving in ways both familiar and strange. In science, the evolutionary history and relationships of organisms, whether an individual species or a whole group, are referred to as their phylogeny. This concept helps us trace how organisms are related through shared ancestry. Phylogeny doesn't always tell us how similar two organisms are in appearance or behavior, just how closely they're related on the evolutionary tree. To map these evolutionary pathways, scientists use diagrams called phylogenetic trees. These trees are visual representations of evolutionary relationships, like family trees for life on Earth. Since we can't go back and directly observe the deep past, these trees are considered hypotheses, models based on the best available evidence. They give us a way to understand how species are connected, when different groups may have evolved, and what their relationships might be. Unlike taxonomic classification, which sorts organisms by shared characteristics, a phylogenetic tree shows how those organisms are related through evolutionary history. Many trees are rooted, meaning they begin at a single origin point, a common ancestor from which all the depicted organisms descended. This root is usually shown at the base or to the left of the diagram. From there, the tree branches outward, illustrating how groups like bacteria, archaea, and eukarya split off and diversified. On a tree like this, plants, animals, and even humans appear as relatively recent offshoots, just a few twigs on an ancient and sprawling tree. Some trees, called unrooted trees, skip the common ancestor altogether. Instead, they simply show the relationships between species without indicating where the group started or how far back their ancestry goes. In rooted trees, branch points represent evolutionary events, moments when a single lineage split into two or more new ones. If a lineage breaks off early and remains separate without branching again, it's called a fribasal taxon. When two lineages split from the same branch point, they're called sister taxa. And when more than two lineages emerge from a single branch point, something scientists call a polytomy, it usually means the exact relationships between those lineages aren't fully resolved yet. It's important to know that rotation at branch points doesn't change the meaning of the tree. You can rearrange the branches at any given node, and the relationships remain the same, just like rotating the branches of a mobile sculpture. The order of the taxa doesn't affect the underlying connections. Phylogenetic trees are tools, and like all scientific tools, they evolve. The field that builds and revises these trees is known as systematics. Systematists gather evidence from fossils, anatomical structures, molecular biology, and DNA sequences to understand how organisms are related. Because new data are constantly being discovered, phylogenetic trees are always works in progress, updated and refined as our understanding deepens. It's easy to assume that closely related organisms must look alike, but evolution doesn't always work that way. Take lizards and rabbits. They may not seem similar at a glance, but both share a trait not found in frogs: the amniotic egg. Despite this, frogs and lizards might appear more visually alike, especially at a superficial level. Looks, as it turns out, can be deceiving. Another common misconception involves branch lengths. On most phylogenetic trees, the length of a branch doesn't represent time, unless it's explicitly labeled that way. The focus is on the order of events, not the duration. So while a tree might show that vertebrates evolved after invertebrates, it doesn't imply that invertebrates stopped evolving. They simply took a different path. And finally, traits can sometimes mislead. Organisms evolving in similar environments might develop similar features, not because they're closely related, but because they face the same pressures. This is known as convergent evolution. When similarity is shaped not by ancestry, but by environment. Let's talk about taxonomy, the science of classifying living things. At its core, taxonomy is about organizing organisms into categories that reflect how they're related to each other. The word itself comes from the Greek for arrangement law, which is fitting, since taxonomy is all about structure and order. If you've ever noticed how a grocery store is arranged, with departments, aisles, categories, and individual products, you already understand the basic idea. Taxonomy works in much the same way, starting from broad groups and narrowing down to specific ones. The system most widely used today is called the Linnaean system, named after Carl Linnaeus, an 18th century Swedish scientist. This system organizes life into a hierarchy, moving from very broad categories to more and more specific ones. At the top, we have three major domains: bacteria, archaea, and eukarya. These domains capture the largest differences among living things. From there, each domain is divided into kingdoms, then into phyla, classes, orders, families, genera, and finally, species. To put that into context, let's consider a familiar example: the dog. A dog is in the domain eukarya, which includes all organisms with complex cells. From there, it belongs to the kingdom Animalia, the phylum Chordata, the class Mammalia, the order Carnivora, the family Canidae, the genus Canis, and the species Lupus. So, its full scientific name, using what's called binomial nomenclature, is Canis lupus. And because dogs are a domesticated subgroup of wolves, they're classified more specifically as Canis lupus familiaris, with familiaris indicating a subspecies. Each level in that hierarchy is called a taxon, and as you move down the list, from domain to species, the organisms within each group become more similar to one another. That's because they're more closely related in evolutionary terms. For a long time, taxonomy relied mainly on visible characteristics, body structure, shape, behavior. But with the rise of DNA technology, scientists now compare genetic information to create more accurate groupings. This has led to some surprising revisions. For instance, while a hippopotamus might look more like a pig than a whale, genetic evidence tells us that hippos and whales actually share a closer common ancestor. Taxonomy today is not just about placing organisms into tidy bins. It's about tracing their evolutionary roots and mapping how they branched apart over time. That's where taxonomy and phylogenetics come together. Phylogenetic trees, the diagrams we discussed earlier, represent evolutionary relationships, and taxonomy helps give those relationships names and structure. When scientists study the evolutionary connections between organisms, they rely on evidence, much like detectives piecing together clues in a case. In evolutionary biology, two main categories of evidence are used: morphological and genetic. Morphological evidence comes from physical form and function, while genetic evidence is drawn from DNA. Generally, the more similar two organisms are in both appearance and genetic makeup, the more closely related they are thought to be. One of the most telling pieces of morphological evidence is found in what are called homologous structures. These are features that look alike and develop in similar ways because they were inherited from a common ancestor. Take, for instance, the wings of bats and birds. Although these animals fly in very different ways, their wings share the same basic bone structure. It's not just one or two bones that match, but an entire arrangement, a layout that's surprisingly consistent, even if the outer shapes and flying techniques differ. This kind of structural similarity points to shared ancestry, and generally, the more complex the shared feature, the less likely it evolved more than once by chance. A simple tool like a hammer could easily be invented independently by two people. But if two people invented the exact same car, with every nut and bolt in the same place, we'd assume one copied from the other. In evolutionary terms, complex similarities are strong clues of a common origin, but looks can be deceiving. Sometimes unrelated organisms evolve similar traits because they've adapted to similar environments or challenges. This is called analogy or homoplasy. For example, both birds and insects have wings, but their wings are built in fundamentally different ways. Insects have no bones at all, whereas birds' wings are bone-supported. So even though they serve the same function, flight, they don't share a common structural origin. These are analogous structures, similar in function, but not in ancestry. It's important to distinguish between homologous and analogous traits. Homologous structures develop from the same embryonic tissues, even if they serve different functions. Analogous structures perform the same task, but evolved independently. For instance, the front flipper of a whale and the human arm are homologous. They share a skeletal structure from a common ancestor. But a butterfly wing and a bird wing are analogous, functionally similar, but evolutionarily separate. Interestingly, some features can be both. Consider the wings of bats and birds. The bones are homologous. Both evolved from a common ancestor with forelimbs. But the wings themselves, the structures used for flying, evolved separately in bats and birds. So in that sense, the wings are also analogous. With the rise of genetic technology, scientists now also use molecular systematics, that is, studying DNA and other molecular data, to understand evolutionary relationships. This has been a game changer. It allows researchers to double-check old classifications and uncover new ones. But it's not always simple. Sometimes mutations in DNA can blur the picture. A few small genetic changes might make close relatives appear unrelated, or distant relatives appear more alike than they actually are. To work around these challenges, scientists rely on computational tools to analyze and compare large sets of genetic data. When morphological and molecular data are considered together, evolutionary relationships become much clearer. The combined approach allows researchers to build stronger, more reliable evolutionary hypotheses. Cladistics is a method scientists use to build phylogenetic trees, diagrams that represent the evolutionary relationships among organisms. Once traits are sorted into homologous, inherited from a common ancestor, and analogous, similar due to convergent evolution categories, cladistics helps organize the homologous traits to map out how organisms are related. The key concept in cladistics is the clade, a group of organisms that includes a common ancestor and all of its descendants. For example, all organisms that have amniotic eggs form a clade because they all inherited that trait from the same ancestor. A clade like this is also known as a monophyletic group. Mono, meaning one, and phyletic, referring to an evolutionary lineage. The size of a clade depends on which ancestor you start from. It could include just a few species or an entire branch of the tree of life. As evolution proceeds, new traits arise through descent with modification. This process usually follows a recognizable pattern. 1. A genetic mutation introduces a new trait in an individual. 2. That trait spreads throughout the population if it's advantageous or neutral. 3. Over time, additional traits emerge as evolution continues. 4. Eventually, these differences lead to new branch points in the evolutionary tree. When examining phylogenetic trees, scientists distinguish between two types of traits. Shared ancestral characters are traits present in a common ancestor and all of its descendants. For example, having a backbone is a shared ancestral trait among vertebrates. Shared derived characters are traits that appear in more recent parts of the lineage and are not present in distant ancestors. An example is the amniotic egg, which is a derived trait shared by mammals, birds, and reptiles, but not by amphibians or fish. It's important to note that whether a trait is considered ancestral or derived depends on the group you're studying. A trait like the amniotic egg is a derived character when comparing reptiles to amphibians, but an ancestral one when comparing mammals and birds. Constructing a phylogenetic tree is a bit like organizing a massive family reunion without a guest list. Scientists must infer relationships based on available evidence, which may include fossil records, anatomical features, and DNA sequences. Things can get tricky, especially when analogous traits mimic homologous ones, making unrelated organisms look deceptively similar. To help navigate these complexities, scientists often rely on the principle of maximum parsimony. This idea suggests that the best hypothesis is the one that requires the fewest assumptions or evolutionary changes. In other words, the simplest explanation is usually the most likely. With the help of cladistics and tools like maximum parsimony, scientists have been able to uncover surprising relationships. For instance, DNA evidence has revealed that humans are more closely related to fungi than fungi are to plants. A finding that would be hard to believe based on appearances alone. As genetic technology continues to advance, our understanding of life's evolutionary web becomes more detailed and more accurate.